Thanks so much for the introduction. It begins in delight and ends in wisdom. Robert Frost referred to poetry, but I think it very much refers to science as well. I like to see my projects like that. I will talk to you about aspects of 3 different projects, all unpublished today, that all sort of connect with each other. There is some machine learning, but it will only be in the last third, I'm afraid. So you have to wait for that. But I'll want to start with actually a tributary slide to Lara and her PhD student Rick, whom I've worked with. So as Lara mentioned, we've collaborated for the last 2 years for a number of projects. And one of them really is going into the nature of lectin binding, where we've combined the human and the machine approach to sort of get a holistic understanding of glycan binding of these complex proteins. We've used a combination of rule-based machine learning and the expert annotation provided by Lara to actually get at these complex constraints and preferences of binding of the 57 most commonly used, arguably, lectins in research. And then later on, really in a project driven by Rick, we've gone to single-cell data. So we've used this unique data set of single-cell transcriptomics combined with the levels of a certain glycan motif on the same single cells to answer 2 questions. So is there enough information in the single-cell transcriptome to predict the level of the single-cell glycan feature? And can we learn from a trained model what features are important for this glycan motif in the case of immune cells and their activity? The answer to both of these questions is yes. So Rick built a neural network that very, very well predicted the level of this feature just purely from the single-cell transcriptome of the cells in a number of cell types here. The model performs very well, and you can use it to interrogate it to find out a lot of the biological associations that have already been uncovered over the decades of experimental research, and we've even found some new postulations there in that regard. But that's sort of, and I very much hope to have even more fantastic collaborations with Lara on that, because I think this combination of different expertises is really what drives the field forward. But I want to go into the full talk here. I think a lot about function. I think a lot, especially in the context of glycans. How do we determine functions of these molecules, which is, as we've heard in the last couple of days, that it's super hard, right? And if you take a look at how people do that in other biological sequences, a very commonly used proxy is conservation. So often, what is conserved is functional. So if you think about genome sequences, if you have a bunch of different bacterial species, you align the genomes, you align their genes, and if you find a conserved gene, a very, very conserved gene, you can be sure that it has some functional role, and especially if you have maybe a catalytic triad that is super conserved, that has a functional role within the protein. Now, you can't really do that for glycans for a number of reasons. I mean, alignment itself is a huge problem. There has been some advances there, how to align nonlinear, highly diverse branch molecules. That is one problem, but that is not the only problem. Because the other problem is that, and that is a major problem in the field as such, I think, information is fragmentary in glycobiology. In the sense that you don't measure the glycome, you measure a part of the glycome. You measure maybe glycoproteins or glycolipids or glycoRNAs, and glycans are in so many different classes. There are GAGs, there are glycans in the ball pit of my daughter. But basically, you measure just a subset, a characteristic biased subset that is not inherently comparable. So if you want to get at conservation, can you compare glycolipids from a donkey liver and glycoproteins from a dolphin brain? Obviously not, right? Because not only is the glycan class different, but also the tissue. So that is a huge issue. You have to compare apples with apples. With bacterial genomes, that's easy because you have the same kind of information from all bacteria. Not the case for glycans. It's even worse because you have subclasses. You can't compare N and O between different species. I mean, that just doesn't make sense, right? You have different ways of releasing the glycans, et cetera, et cetera. So there's just so much bias, so much fragmentation that it's tough to deal with that. So we thought about what would be a showcase to actually compare apples to apples. And in our case, we decided to go with milk. So breast milk, all mammals produce breast milk. They contain also glycoproteins and glycolipids and whatever, but they contain another class of glycans, is free milk oligosaccharides. So soluble glycans that make actually quite a sizable fraction of this liquid and have a lot of functional relevance for breast milk. So they all stem from lactose with a bit of an asterisk that I don't go into, but then get elongated by enzymes. And it's really cool because they all come from the same tissue, they all come from breast milk, they only occur there by definition. They're the same class. They don't have to be separated, so you still sort of have to extract them a bit, but you don't have to actually cleave them off something because they're already free. So you don't have heterogeneity in that sense. It's also a quite non-invasive sample. You don't have to cut something open or whatever to get breast milk. It is available in relatively high volumes compared to some other precious samples. Again, breast milk glycans are super important for a lot of different functions. So anti-pathogen functions, nutritional functions, developmental functions, etc., that are tied to specific motifs, to specific glycan sequences. And yeah, that makes them really worthwhile to study, I think. Just if you go into a bit more detailed examples of what are their anti-pathogen functions, one example is that they can actually bind to pathogen lectins, so by mimicking host epitopes basically, and thereby preventing bacteria, viruses, etc., from binding to the mucosal surfaces. Another more recent discovery is that they can actually disrupt biofilms, so they are of interest for antimicrobial resistance efforts. And even more recent, the Townsend lab has actually found that you can use them to co-treat bacteria with antibiotics. So they sensitize antimicrobially resistant bacteria against antibiotics. So there's a lot of biomedical relevance also in these molecules. I'm pretty allergic to dogma, so another reason for me to go into this is that I heard left and right that humans have the best, the brightest, and the most complex milk oligosaccharides, and I'm always a bit skeptical when I hear things like that. So I went into, and I'll go into our data collection efforts later, but I actually correlated the number of publications on a species with the number of known oligosaccharides, and it's a really strong correlation. So I think it's mostly just a sampling bias. Obviously people want to study humans, I totally get that, and therefore we know a lot about human milk glycans. We actually know about close to 300 unique milk oligosaccharides from human milk. That's definitely an outlier by all means. But for most mammalian species, we have actually no information at all about their breast milk glycans. What is true, and I give people that, is that humans tend to have higher levels of milk oligosaccharides. That makes it a lot easier to study low abundance species, of course, because you have to remember you measure these glycans via mass spectrometry, so sensitivity is an issue, and that is easier in human breast milk. But we don't have really a clear sense of what is actually the comprehensive diversity over the whole class of mammalia. So what is lurking out there? We have an overrepresentation of human and model species like domestic cows, sheep, milk, etc. But in order to really get a sense of what is conserved, what is functionally important, we needed to get a sense of what do we actually know, because there is no centralized repository of known milk oligosaccharides, something like that. So we actually went into the last 120 years of milk research. It's a lot of thousands of papers. And we cataloged all known milk oligosaccharide structures. So as you can imagine, there's lots of artiodactyla and primates, artiodactyla, cows, sheep, goat, etc. Primates, mostly humans, to be honest. But we have information to some extent from over 100 mammalian species. That being said, most species are like 2 to 5 milk glycans that we know about. And those 2 to 5 are always the same. It's like 2-fucosyllactose, 3-sialolactose. People discover them over and over again in different contexts because they know how they look like, they know what to look for, so they just have to recognize them and then tick off the box, basically. Just here as a caveat that for actually, at least for humans, we have data from thousands and thousands of individuals. Most animals are a single animal, so we don't even know if that's an outlier for the population or not. And here's just again the emphasis on the emphasis of researchers on these simple, abundant, but of course relevant sugars. But we don't know exactly what else is out there. So working with data, I find the easiest way to understand data is to generate data myself. So that's exactly what we did as a next step. So we have the literature-generated data. So those are around 1,700 observations of structure A and species A. But we wanted to add to that. We wanted to understand what are the limitations, what is the actual diversity in samples if you go deep, if you invest the time and the resources to find out what these species actually harbor in terms of their structures. So we did that. We collaborated with a lot of zoological institutions in Europe to get samples, because sample availability is a key issue if you want to find out new species and new milk glycans. We have milk of one primate. This is Luast's monkey, and the number here indicates our current glycobiological knowledge of this little monkey, which is 0. Most of these species are complete frontiers in the sense of our knowledge of which glycans they exhibit. don't get me wrong, they all will have man9, glycan2 or whatever, but they will have, all of them will have unique biochemical properties which are worth studying, I think. We also have the black rhino here as a Parasodactyla representative. We have a bunch of Artiodactyla. We are also, we contribute to the problem of a bias of Artiodactyla species just because there are so many of them that are relatively easy to get. But there are some cool ones here, including the aquatic ones. I really recommend the pygmy hippo. I've seen them in the Toronto Zoo a couple of weeks back. They're really cute. Love them. And then as a last-minute addition, we added sheep milk as a sort of example of a more widely studied animal to see are the findings that we have in these other species an outlier, or are they also recapitulated in a more well-known species. So we got all of these milk samples from these animals, and then we went into what I would almost call deep milk glycomics, especially looking at the deep hole in my budget after all this. But it's basically negative mode mass spectrometry. For the aficionados, we went to MS, so to really fragment all the way down. And when I say we, it's mostly Jin Cheng from our core facility, because I have no idea about mass spectrometry, and he has all the ideas about mass spectrometry. So we have a great core facility at the University of Gothenburg for glycomics, so we work with them, with this national infrastructure BioMS. And it's really going all the way with exoglycosidase digestions and et cetera, to get at all these structures. So there are a lot of structures, and we're still in the process. This is still an ongoing process, but we already uncovered more than 70 and characterized, really, to the last detail, more than 70 structures that are completely unknown. To our knowledge, completely novel, with some really interesting properties, and I'll just highlight 2 of them in later slides. And here are just some MS1 scanned examples. I go into a bit more detail in a second. But I want to just highlight what I forgot here is that this already is basically over 12% increase of the total known unique structures that we know about as free milk oligosaccharides. And just by taking 8 or 9, depending on how you want to count, randomly chosen species. So just shows you how much undersampling I think we've done so far. Here in the bar chart, you see in blue the number of characterized structures for each species. In orange are the number of completely new structures from this species. So some of them, like the black rhino, excellent. 2/3 of the structures are completely new. Amazing. And this is already outdated. We discover new ones basically every couple of days as it's still ongoing. So it's a gift that keeps on giving. We didn't want to lose our complete sanity, so I didn't draw all of them. I just drew a couple of them here. One of my favorites, top left, 6-sulfo-2-fucosyl-lactose. Very unusual and not seen before. It's a modification of a super well-known milk oligosaccharide, 2-fucosyl-lactose. It's speculated that some form of it exists in humans, but it has never been shown. It definitely exists in dolphins, though. I want to highlight 2 examples. One is lacdinac as a motif. Lacdinac here, galnac beta-1,4 glycnac. As a terminal motif usually. We find it in a lot of species actually. We find it in 2/3 of our species. To our knowledge, this has not been reported in milk glycans before. It has been reported, of course, in other mammalian glycans. Don't want to claim that. Here, for instance, is an example of a pygmy hippo glycan that not only has a lacdinic motif, but also an alpha-gal moiety. So quite cool molecule. In other glycan types, it has been reported in N-glycans, in O-glycans, and probably in glycolipids. But what is really cool here, if it is salicylated in these types of glycans, it is usually salicylated on the galnac. What we find is salivation on the glycnac residue. Actually, here are some examples of lacdinac-containing milk glycans in some of our species, and they look quite different. So it will be interesting to dig into their biosynthesis. We are currently looking into the functions of this particular motif. Just as a teaser, lacdinic glycans are quite prominent in parasite glycans, and we know that galactin-3 actually binds lacdinac and uses it for immune recognition. So they could very well have a similar sort of immune educating or even potentially antipathogen function in the milk. Where we do have some functional information is for the other motif that I want to briefly talk about, and that is actually glucuronic acid is a new monosaccharide in milk glycans. So we are not the complete first to identify this, but we are the first to structurally identify it. Here is an example of I think one of the dolphin glycans actually that contains this. If you are very mass spec affine, you might appreciate the ion at 619, which is the loss of a carboxylic acid, and can distinguish a methylated hexose from the actual glucuronic acid, which are isobaric otherwise. So we can be sure that it actually is a glucuronic acid. Also of note here is that it's actually quite unusual to have an internal glucuronic acid in a glycan, in a non-GAG glycan. So in glycosaminoglycans you have it per usual internally, but not in non-GAG glycans. So very unusual. And we find it in 5 of 9 species, so it's not an outlier. It's actually present in more than half of the species that we investigated. So it seems to be quite conserved, actually. Last year, the Zandberg group at UBC first reported glucuronic acid-containing milk oligosaccharides in cow milk, but only at the compositional level. So they did find several trisaccharides here with glucuronic acid. And some of them seem to be even sulfated, which is actually quite cool because we know, I mean, here none of the sulfations are indicated to be at the terminal site, but we do know that even in humans we have the HNK1 motif, which is a sulfated glucuronic acid, so CD57 here, that is present on other types of glycans. And that is important in a lot of different functions. So it will be intriguing to see whether the glucuronic acid in milk glycans maybe has also some functions that we don't know yet. What really helped us here is that sheep milk actually has a ton of it. The acidic fraction, so you just get the acidic milk oligosaccharides. More than half of the entire acidic fraction of sheep milk glycans are these glucuronylated milk glycans. So don't ask me how people have missed that before, but apparently they did. But that is really great because that is a really enriched fraction that we can use for experiments. The other part of it is cell lactose, so we can actually sort of disentangle that. So what we did there is we took differentiated THP1 cells as sort of a stand-in for human macrophages. We stimulated them with LPS or not, and with the different milk oligosaccharides to see if there is an immunomodulatory effect. Just for context, glucuronic acid also is a parasite motif, so it could also be in the same direction of the lactinic, but because of the HNK1 motif similarity, etc., we thought that there could be some immunomodulation going on. And then we just assay this with a cytokine multiplex assay to see what happens. And of course, LPS brings up all the cytokines. That's no surprise. We have 2-fucosyllactose as a sort of immunomodulatory positive control, bringing down some of the cytokines. We have pure glucuronic acid mimicking the effects of 2-fucosyllactose, so pure glucuronic acid by itself already has an effect. The acidic fraction of sheep has an even stronger effect, which you do not see in the cellular lactose case. So there's a strong indication that it actually stems from these glucuronylated milk oligosaccharides. And by now we should have actually gotten chemically synthesized elongated glucuronylated milk oligosaccharides to test that even more conclusively. But I want to go back to the beginning, to conservation, to this idea of conservation. So this was sort of our functional foray into some of the discoveries that we made, but we initially wanted to do that in order to get at glycan conservation in this constrained class. In order to do that, we have to make methods. It would be remiss of me to do a talk without mentioning GlycoWork, or open access Python package that we use for everything glycobiology. And that we also use for this. So we added a new module to GlycoWork that's called Network. Network has 2 submodules: biosynthesis for constructing and analyzing biosynthetic networks of glycans, and evolution for assaying their conservation. And we did a lot of those. I only can talk about a few things that we did, but just to prime you, the idea of a biosynthetic network is really cool for milk glycans because, again, they mostly stem from lactose, so that is sort of the biosynthetic route. These are the alpaca milk glycans as a toy example because they're relatively tame compared to some of the other species. In green, you have observed milk oligosaccharides. In pink, you have intermediates that are not observed, but that could conceivably connect observed milk oligosaccharides. So this is just the starting point, the sort of brute force. How can I force this network to be connected? Because we know it has to be connected. Milk glycans don't appear from nowhere. They have to come from something, biosynthetically speaking. And every step, and an enzyme adds a monosaccharide or a modification as a step in this network. So every step is a reaction, basically. And already here you can make some interesting observations. One observation that we already made by eye is that we have some hubs. The nodes are scaled by the number of connections that they make. So you have some hub molecules in these networks. And what is interesting and what we sort of more or less manually already identified is that these hubs tend to be actually unobserved intermediates. You can quantify that across our 100-something species. On average, it indeed tends to be that unobserved intermediates are hubs. And the reason for that is they're hubs by definition if they have a lot of arrows that are outgoing. So they are bottlenecks in reactions in a sense. They are being metabolized by a lot of different pathways, and therefore the levels are conceivably too low to be identified in most routine mass spec endeavors. And you can have a more comprehensive look at that by actually looking at all the 100+ networks of the milk glycans of mammals to see which milk oligosaccharides tend to be unobserved, which tend to be always observed. Here are some of the more extreme cases. On the bottom, you have the ones that are always observed. Those are terminal glycans, so 2-fucosyllactose or lactose and things like that. Those do not get extended, so they're done. They just accumulate to some extent, and then you can measure them. Whereas on the top, you typically have a strong enrichment actually for glycnac terminated milk oligosaccharides, so sort of like unfinished lacnac repeats, if you want to phrase it like that. So there seems to be a very efficient draw into a completed lacnac, which we observe here, and these intermediates are not really observed, but are necessary because you can't have a lacnac repeat without first having the glycnac, right? And that really led us to this idea of also using that to inform our networks. Because if you have this toy example here of a diamond structure where you have a glycan at the beginning and glycan at the end, and you have two different additions that you have to make, not contiguous, but at different positions. There are two possible routes, right? So you can first add the galactose and then the salic acid, or first the salic acid and then the galactose. And it's not random which you add, because these are enzymes, they have specificities, they have substrate specificities, they are evolved, so they likely maybe even are conserved in terms of their specificity. So in this case here, for instance, in 2/3 of the cases where we see this path in our networks, it has experimental support, and in 0% of the cases, this path has experimental support. So it is, and this is actually the usual case, that one path is clearly favored across 100-something species. So there's a strong conservation even across the whole class, which really allows us to prune the network, because we can prune away. So for instance, here you see that there is a diamond structure where even in alpacas one of the options is observed, and the unobserved version is pruned away. And that already gives you a much cleaner network. You can prune that further with various other evolutionary strategies that I won't go into. But what is also interesting is that this makes the hubs even worse. So there are even more hubs that are necessary for the network. And we did a lot of biosynthetic modular analysis on that, which I won't go into. But what we did really go further into is this idea of path dependence of reactions. So that you need to have a precursor, a sufficient precursor for the enzyme to actually act upon, and to see, is that generalizable across glycan sequence contexts? So, for instance, here in this case, it would be just abstract the idea that you have a galactose beta-1,3 addition and a salic acid alpha-2,6 addition. In this context, it's obvious which direction occurs. What is it in another context? You can go across all glycan contexts in our networks and actually see, are there generalizable rules about this? And there are. I mean, these are the most extreme differences, basically, just fished out, basically. But you see that there are clear orders of differences, and they're thresholded, so they're not rare, isolated cases. So they're only cases that are sufficiently frequent in our networks to actually make educated conclusions about that. And what is really interesting is that not only, so this is now across glycan sequence context, but also you see that there are even similarities here across different reactions. So for instance here, both salic acid orientations here come after galactose addition. And remember again, this is not in line. In line you only can have one order, right? But at different positions in the glycan. There is a caveat here that we see some differences between taxonomic orders depending on the reaction, but a lot of these are really conserved across whole mammalia. So there's a lot of conservation in the specificities of the enzymes that are going into determining which repertoires of glycans you actually have in your milk. And if you want to be cheesy, you can make sort of a hierarchy of which monosaccharides are preferred in terms of the addition order here. Okay, so and like I said, we did lots more, but I want to go into the last third here, and that's so. This is a really fun project. I love it, and we learned a lot about it. But it has at least one downside, and that is that it took so long. we're still not finished. I mean, it's nine samples or nine species, right? And we started a while ago, and we are not finished. It took a lot of time, and we have we actually we have more samples lying around. But that will be another project because I want to get this out and I don't want to spend another. I want to retire at some point. No, but there are a lot more species out there. Like I said, we have around 6,500 mammalian species that we know about. So if we want to get even sort of an inkling of what is out there, we need to accelerate this. And as anyone that really does detailed mass spec will tell you, is that the analysis part is the one that is driving the time commitment here. I mean, I've exaggerated a bit here, but in any case, it's really the detailed, exoglycosidase digestion and going into the fragmentation, etc. To figuring out what is actually the molecule is the part that is giving us trouble. So we thought about how to accelerate that and to giving us an ambitious goal, how to bring that from months to seconds. And we do machine learning, so why not do machine learning? So this is why we thought about doing Candy Crunch. So Candy Crunch is our deep learning model to predict glycan structure from MSMS information, or actually LC-MSMS information, which is not meant to really replace the analyst, but more supplement the analyst. It's a human plus machine approach as before. So to accelerate, because there are lots of steps that can be accelerated, I believe, and then together with the expertise of the human can be perfected, hopefully. But in order to do that, it's a very hard problem. So for every hard problem, you need lots of data. So this is actually, I think, the most data-hungry project that I've done ever. So we've tried to gather all of the mass spec information that is out there. By the way, if you have mass spec information, please either upload them or give them to me. Otherwise, you have no right to complain about insufficient performance of our model, just wanting to put out there. But lots of people do, so that's great. So lots of our data is actually from Glycopost, so that is a really good repository, which we have gone through completely. We've also gone through completely all of the other resources here. And to get here terabytes of raw data in the end, and information from a lot of different glycans. We have the same biases as the field has, or at least as the people that deposit data have. Animal samples are definitely the majority. There is a bias towards negative mode mass spectrometry. There's a bias towards protein-linked glycans, by far. So our milk glycan information actually helps it to rectify that a little bit. But yeah, so we have the data basically. We have sort of the model sketched out and working. So it's a quite complex model. It takes both the information, the actual fragmentation and the tandem mass spectrometry. So how does the molecule fragment, and information from that. But it also takes tons of experimental setup information. So the retention time of the liquid chromatography, which liquid chromatography did you use, how did you modify the glycan, etc. All of that is learnable and included in the model input, and really helps in figuring out what the structure is. In the end, you get sort of a probability list of structures, so that's really useful for the analyst in the end, that they can rule out things that they know are not true, but at least still gives them sort of maybe as a best guess. What is even more important, I think, than the model is the actual workflow afterwards. So remember, we do everything in Python. We can't rely on any peak calling or I don't know, a spectra picking or something like that. We have to implement everything from the ground up. So the actual workflow after the prediction has maybe taken me even more time to do than the actual model itself. Because the model works on single spectra, but what you do want to work on are whole samples. So raw files of whole samples, which are thousands of spectra, because they're biosynthetically related, and because you don't know a priori which maybe is the highest quality spectra. So what we do is we just predict all of them, and then we filter down. So we basically have a raw file. We predict the glycan for each spectrum. We throw away a lot of that. So we of course, you have for the same glycan maybe multiple spectra. We only take one right from the middle of the distribution. So mass isomers, retention isomers, we have we deal with all of those. We have a lot of domain filters to filter out easily checkable unphysiological predictions. So for instance, just a random example, if you negative mode predict a cell-related glycan, but you don't have a 290 peak or a mass minus 291 peak, then likely not true. So things like that, just to avoid or minimize the number of false positives. And it's really, really fast. I mean, the prediction itself actually takes not even a second or something. The workflow actually takes the majority of the time, to be honest. But it's still, it takes less than a minute per sample. And just to flash you with some numbers, so this is sort of how we're currently doing, broken up across different levels. So the total accuracy at the moment of structural prediction is around 82% accuracy. That's top 1 accuracy, so that the actual top 1 prediction is correct. And it gets better, of course, if you have more granular information. What is important to note, though, is that this is the actual accuracy of the model output. The workflow improves that, because the workflow rules out mass errors, rules out all kinds of unphysiological predictions. So the accuracy that you get at the end of the workflow is higher than the pure model accuracy. And yeah, and then we work with actual mass spec people to cross-check these things, etc., to help us build that. And so if you just want to visualize here, these are 15% of our spectra, so like, I don't know, 30,000 spectra or so, plotted and colored by glycan class. What is really cool here is you see these streaks. Those are spectra of the same glycan molecule, but under subtly different experimental conditions. So we have sort of replicates from different people, different setups, etc. But this is a learned representation, so closeness implies similarity. So the model has correctly learned that this is the same thing, just subtly different, so it puts it together. And what you can do now with that is really fun, because now you can do really high throughput analysis. We have, like I said, hundreds of data sets that we curated for this model. So we can actually, for instance, analyze all glycomes that we know about. And that's, like I said, it's hundreds of thousands of spectra in that case. So we can, for instance, take I should mention the benefit here that I find is that everything now is processed the same. So the raw file, we take the raw files of everyone, and the model has its biases, for sure, but it's the same bias for every sample now. So that's good, I think, because now we can take, for instance, here just as a toy example, we can take human tissues and just get the different distributions of cell-related, fucosylated, or fucosylated glycans in different human tissues. Clearly seeing here brain cellulation, stomach fucosylation, things like that. Just sort of as a sanity check. What we also can do is do the same thing that we did for the milk glycans, do network analysis. So here we just build networks from the O glycomes, which should also be biosynthetically related, and do the same thing, reaction order dependence, for instance, where we actually surprisingly see quite a similar hierarchy of monosaccharide additions than in the milk glycans. No idea whether that's true for the other classes as well. I haven't actually checked yet, but it would be interesting if there are actually more general biosynthetic constraints than just within one class, which makes sense because it's a bit artificial to separate it into classes anyways. And it's, again, the same observation that a lot of the intermediates are never observed and are critical hubs, so are basically used as a bottleneck in biosynthesis. Let me go to the summary. So the first 2 projects are basically more or less submission ready. The Candy Crunch, not quite yet. I think we're quite far there, but still need some tuning. What I find really encouraging is that when we work with mass spec experts, we actually get also really good predictions from not so good quality spectra and from single scan spectra, which actually many analysts even discard because it may not be so robust to actually get an answer out of that. So that's really cool, and we are really excited about both the scale that this may allow for, as well as the acceleration, especially, I mean, even egoistically in our work, to accelerate our sample measurements would be already worth it for that. And so, like I said, we are continuing with our mixed studies. We have lots of species that we already have available. We have plans for more. Especially because we see these conserved novel motifs even across mammalia that have been flying under the radar for now. By the way, before you ask, I have no idea whether they occur in humans or not. To our current knowledge, they don't occur, and people have looked extensively. So that's actually kind of strange. But we didn't analyze human milk so far, so we don't know. And I want to end with a quote from Louis L'Amour. So when you think that you are at the end of the whole thing, then you're probably just at the beginning. And that's another encapsulation of research, I think. So with that, I want to thank, first and foremost, actually, our zoological collaborators. Without the samples, a lot of this would not have been possible. But also our collaborators, our funders, and you for your attention.